It's a great pleasure to introduce you another speaker today. Tom Scanlan is a professor of physiology and molecular biology at Oregon Health Science University. Yeah. His background is in Thyromimetics, and this is an interesting class of drugs that was used in the old days for people to lose weight and reduce their cholesterols, but we're now finding that it may have interesting implications in the disease that we're interested in. So I'm just going to talk. Thanks.
Is this working?
Yep.
Okay. Well, thank you, Ollie, and Florian for the invitation. My background is Thyromimetics and metabolic disease, weight loss, obesity, diabetes, and hypercholesterolemia. And so, but I have immersed myself mostly in a vacuum, but occasionally with other people over the last 2 years in ALD. I hope to show you a connection between this class of compounds and ALD. And it's just great to come to this meeting because just like someone learning a new language, conversing with people about ALD and that kind of thing here, it's really about the first time I've done that. And I can, as was mentioned yesterday in the endocrine section in the afternoon, there's really no discussion of ALD. You'll get blank stares from the average endocrinologist when you bring it up. And maybe if you say adreno or Addison's or adrenal insufficiency, they'll perk up a little bit, but not when it comes to the neurological involvement. And just also by way of disclosure, I'm a founder of Neurovia, which is a seed stage company that's pursuing one of the thyromimetics that's published on probably the most, Sabet Orem. This analog or agent was discovered in my lab in academia. But nevertheless, what I want to do is make this talk informational and conceptual about the area, just thyroid hormone action and what that can do for ALD. And I tried not to make this an elevator pitch for Neurovia. Okay. And so let's start with bringing everyone up to speed with the thyroid hormone axis. The gland is located right here at the base of your neck, and this is and it produces thyroid hormone, thyroxine or T4 right here, which has 4 iodines in it. That's why it gets its name T4. And this is the reason you need iodide or elemental iodide in your diet. And the only reason you need it is to make this hormone. So the gland takes up iodide, combines that with tyrosine residues, makes thyroxine, which is the major form of the hormone secreted from the gland. Interestingly, though, and so that all through blood, this is what the main form of thyroid hormone is, but this is not the active form of the hormone. It's really a prohormone. And what happens is thyroxine is taken up into target tissues and target cells within target tissues. And as you'll see, virtually every tissue is a thyroid hormone target tissue. And once taken up, it is then converted to T3, triiodothyronine, by specific enzymes called deiodinases, and there are 3 of them. And so you can see that one of these iodides has been knocked off. And so this is the form that binds to the thyroid hormone receptor and has thyroid hormone activity. So like all hormones, blood levels of this have to be tightly regulated, and they're done through the hypothalamic thyroid pituitary axis. And that involves secretion of this factor, TSH, or thyroid stimulating hormone, which is basically a sensor that determines how much of circulating T4, T3 there is in blood. And so when T4 levels are low, or if you're hyperthyroid, that drives more secretion of TSH from the pituitary, which instructs the gland to make more T4. And conversely, when T4 is high, or hyperthyroidism, that suppresses the amount of TSH that's secreted from the gland. So the way thyroid hormone works mechanistically, it's in the family of the receptors, the thyroid hormone receptor. It's in the superfamily of nuclear hormone receptors, which basically are ligand-activated transcription regulators. So they're soluble proteins. In the case of thyroid hormone receptor, it's located in the nucleus. And what it basically does is controls the transcription of thyroid hormone responsive genes, of which there might be hundreds of in any given cell. And so a thyroid hormone responsive gene contains in its promoter a DNA binding site, a thyroid hormone response element that binds the thyroid hormone receptor. And then the hormone secreted from the gland goes into blood, makes it all the way into target cells, into the nucleus, where the receptor is, binds to the thyroid hormone receptor, and in the simplest positive regulatory case, that turns on or transcribes, increases transcription of the hormone responsive gene. And so it's interesting, thyroid hormone has to be in balance by the two slides that I just showed you. And so it does it because it works on all of these different things in all of these different tissues. But it's interesting from a therapeutic development standpoint to consider the clinical presentation of hyperthyroid individuals. Because what you see when you look at that is there are good things that thyroid hormone does in that disease, and there are bad things. And so, for example, hyperthyroid people are always thin, or pretty much always thin. And that's because their metabolic rate in all cells is boosted up. And so they're burning calories faster, and thyroid hormone does that, and they're burning calories faster than they can take them in, and so thus they lose weight. The second positive thing is that LDL cholesterol, which is a marker of atherosclerosis, cardiovascular disease, is rock bottom low in hyperthyroid people. And thyroid hormone, in fact, is the most potent agent known at lowering serum cholesterol, and it does that by controlling cholesterol clearance through the liver by upregulating a number of mechanisms. One involves upregulation of the LDL receptor, which basically just clears out LDL cholesterol from serum. And so those are the two good things, but the problem is hyperthyroid people have increased heart rate and contractile force, which is dangerous in the short term, putting that extra stress on the heart. If they go long enough, they will have osteoporosis because of the effects that thyroid hormone excess has on the bone. And in some cases, they have psychiatric symptoms bordering on psychosis. And so in extreme cases, I would say. And so these are the negative things. And so thyroid hormone in excess doesn't distinguish any of these, but the drive of thyromimetic development is, can you make synthetic thyroid hormone analogs that do the good things about this and not the bad? And that's basically been what the last 20 years of my career has been spent doing. So why would you consider a thyroid hormone agonist or a thyromimetic for treating XALD? And there are really two mechanisms, and one is fairly straightforward, and most of you probably already know this because the discovery of this came from the ALD community, not the thyroid community, or the endocrine community, and that is that the gene ABCD2, which is 70% identical in protein sequence to the defective allele in ABCD1 or in ALD, ABCD1, is regulated by thyroid hormone. So it has a classic positive regulatory thyroid hormone response element in its promoter, and therefore T3 will turn this gene on. And it's been shown that if you can figure out ways to turn this gene on, it can compensate in terms of the biochemical phenotype, degrading VLCFAs for defective ABCD1. So that's the first mechanism. The second mechanism, which you may not be as familiar with, is that thyroid hormone plays an absolutely critical role in myelin development and in myelin repair. And the way it does this, the ways they're understood at least that it does this, is it drives, in development, it drives the differentiation of oligodendrocyte precursor cells into oligodendrocytes, mature oligodendrocytes, which are then the cells that go and lay down myelin on developing neurons. But there's also, in addition to this developmental aspect, there's a growing body of evidence just in the last few years that thyroid hormone control, there are certain genes that are thyroid hormone regulated within mature oligodendrocytes that are responsible for repairing demyelinating lesions in adults. So developmental myelin role also repair. And so let's look at some data to support both of these. And the first comes from published data, and I show this just to bring it back to the idea that this really came out of the ALD community. A French group really, as far as I know, formed the first connection between thyroid hormone action and ABCD2 transcription. And then the Austrian group followed up on details of that. And then later, this same French group showed this data where they these are XALD fibroblasts, and they show that T3 turns on transcription of ABCD2, and they also interestingly showed that thyromimetics do the same thing. And this caught my eye because I'm in the thyroid community. I totally missed this publication. It was years later that someone mentioned this to me, and I knew nothing about ALD. And my first question is, how did they get their hands on Sabet Orem? Because I thought at that time I was the gatekeeper of all the Sabet Orem in the world. But I guess you never are. It's never as good as you think it is. And they did get it, and showed that it turns on this ABCD2 gene. And we've done some follow-up things. This is unpublished work from my lab to show that this also works in vivo. These are mice, wild-type mice, that have been treated. We either make them hypothyroid by giving them a thyroid hormone biosynthetic inhibitor, and that takes about a month or so to work, where they're clinically hypothyroid, or we inject daily thyroid hormone T3 into the mice to make them hyperthyroid. And what we show from this is that ABCD2 in vivo is thyroid hormone regulated also in affected tissues in ALD. And so the liver is not one of those tissues, but you can see the classic signs of a thyroid hormone regulated gene. It's upregulated in the case of a hyperthyroid animal, and it's downregulated in the case of a hypothyroid animal relative to the euthyroid control. And then in whole brain, we see extracts from whole brain. We see the same thing. This is qPCR, just of these, of whole brain extracts. And then in a white matter-rich region of the brain, the corpus callosum, you also see this same thyroid hormone regulation of ABCD2. Now, we've also shown that the circulating biochemical phenotype biomarker of ALD is also responsive to thyroid hormone control. And so these are ABCD1 knockout mice, which we've been spent about the past year or so developing a colony in my laboratory. And as has been reported, they have elevated circulating VLCFAs compared to wild type. We're measuring here total C26 by the GC, the older GCMS method. We also use the C26 lyso-PC assay, and the results are identical. We don't see any difference between those two. And what you see is that when you make these ABCD1 knockout mice hypothyroid, that circulating C26 goes up relative to the euthyroid control, and likewise when you make them hyperthyroid, it goes down relative to the euthyroid control. And so, there are different ways you can interpret this, but I think at least some component of this has to be activation of peroxisomal degradation of the excess very long-chain fatty acids in these mice, depending on thyroid status. And so what about the remyelination aspect of thyroid hormone? There's actually lots of data to support this, and I'm just from many different labs, and I'm just showing you one such paper published in 2008. And what these people used is this Cooperzone model that, mice in the Cooperzone model. So Cooperzone's a copper chelating agent that creates demyelinated lesions within the brain. And myelin, you can see here, before the Cooperzone treatment is stained in red. That's for myelin basic protein in the corpus callosum. And then you can see the red going away as you treat with Cooperzone. And then if you take these animals and treat them with T3 versus no treatment or vehicle, you can see a repopulation of the red, or an increase in myelin content within the corpus callosum. Another way of looking at it from the same study is EM, and so you can see here these are normal myelin sheaths, cross-sectional view of them from the brain. Treat with 12 weeks of Cooperzone, and you see that those go away. And then without T3, you get some spontaneous remyelination in this model, and just about every model of demyelination. In mice, or in rats, there's spontaneous remyelination. But with T3 treatment, you see an increased level of recovery of these myelin sheaths. And so a demonstration of remyelination. And then interestingly, these researchers did a clinical test. This is a rotor rod showing tests for these mice, showing the behavioral clinical phenotype decreasing through Cooperzone treatment, not really increasing very rapidly in the control animals, but increasing more rapidly to normal in the animals that were given T3 over that period. And so then you're asking yourselves, or I would at least, if I wasn't a thyroid hormone aficionado, why don't you just treat with thyroid hormone? I mean, thyroxine is a generic drug right now. It's approved. Why wouldn't you just give patients thyroxine? And the reason is back to that second slide or so that I showed you, is there are these thyroid specific adverse effects which really preclude you from doing that. In the short term, the thing we worry about the most are cardiac effects, but then over a longer period of time, you've got osteoporosis, you've got muscle wasting, which is also a disease, and then the psychiatric symptoms that might develop with high doses and prolonged treatment. And, it's not that people haven't been thinking about that for a long time. They've been trying to use thyroid hormone for something other than thyroid hormone replacement for decades, and it frankly just doesn't work because there's no therapeutic window with thyroid hormone. And this is an illustrative study of this. This was 1972. Cardiologists knew at this time that LDL cholesterol was a problem and a marker for atherosclerosis, yet there weren't any statins or any of the drugs available today to lower it. And so they embarked on a study in the U.S. to study thyroxine and other agents, to try and get therapeutic benefit from them. And it's called the Coronary Drug Project. And what you can see with thyroxine treatment is everything's fine for about a year or so. These are once daily doses of thyroxine that are mildly hyperthyroid type of doses. And in all of these patients, the LDL cholesterol went down. There's no doubt about that. But then after about a year or so, this is T4 and this is placebo, and this is a measurement of mortality on the y-axis, and you can clearly see that there are drug-induced deaths that creep in after about year one. And so as the ultimate safety endpoint of something you can't breach, mortality would be that. You can see that this is simply not safe. It's not safe to make patients hyperthyroid, globally hyperthyroid. And so that's why thyromimetics had to be developed, or the question was, can you tease apart in a tissue and possibly receptor specific way? There are actually two thyroid hormone receptors. I didn't tell you that, TR alpha and TR beta. But can you, through mechanism or tissue selectivity drug uptake, get a selective response or a therapeutic window with the thyromimetic versus T3? And this is a nice illustration of that you can. The answer is yes. And these are rats. They're hypercholesterolemic rats that are treated once daily orally for 7 days with either thyroid hormone T3 or a thyromimetic drug, Sabet Orem. And what you can see in the T3 case, we're measuring here heart rate in that direction, cholesterol, which is the positive therapeutic endpoint we want to lower in that direction, and then in black, thyroid stimulating hormone, or TSH, which is something you don't want to change. And you don't want to perturb the thyroid hormone axis. And what you can see with T3 treatment is there's no therapeutic window. At every single dose where you get cholesterol lowering, you get a heart rate increase and you get TSH suppression. And so there's simply no therapeutic window with the natural hormone. But with Sabet Orem, you can see that you get cholesterol lowering, good cholesterol lowering, and there is a window. There are doses that lower cholesterol fairly well, have no effect on TSH, and also have no effect on heart rate. And so that was really, this was the excitement really that led to a number of different activities in industry. And I guess I'm one of the only academics that got involved in this, but in trying to develop these agents as cholesterol, as drugs that would lower cholesterol. And this really started in the mid-90s, and when I started my career and went through up until, I guess, recently, but a lot of activity in the early and mid-2000s. And what I'm showing you here on this slide are the clinical stage thyromimetics that are out there right now. So these are the ones I know about, and I don't think there are any others, but someone can correct me if I'm wrong. And these are the compounds for which clinical trials have been done in humans. So these have had at least at one time an active IND, some still do, and there's been at least phase 1 studies reported also in humans. And the one where the most work has been done is this compound of proterome, KB2115, and it was produced by a Swedish company, a small nuclear receptor Swedish company called Bio. And they got all the way into a phase 3 before they had to stop that program. And I'll show you a little bit of data from that and talk about why they stopped it. This is the compound developed in my lab, and the reason it made it into clinical development, this was before NCADs. There wasn't anything like that available back then, so for an academic scientist to try and get an IND, that's the valley of death that somebody was referring to yesterday. And the problem is it costs $3 to $5 million, and that for starters, and that isn't available usually through grant funding mechanisms. But a company called Quatrex, which is barely around anymore, they might have a website, licensed this. They were interested in developing cholesterol-lowering medications with new mechanisms, and they licensed Sabet Orem from my university. And now it's being pursued by Neurovia. So they're all, which is NV1205. So there are all these different names, and we originally called it GC1. And so there are all these different names for it in the literature if you run into that. And this one is an interesting compound. This is from Metabasis, which they had a different strategy. They really wanted the liver tissue selectivity for their compound, and they'd give it orally. And so they put this bit over here on there. I am a chemist by training, actually, so this all makes perfect sense to me, but I know that some of you it probably doesn't. But don't worry, we're not going to have a chemistry quiz or anything like that after this. But if you have a question about it, just let me know. But all of this stuff is to mask this ligand so that it will not bind or activate thyroid hormone receptors. So it's called a prodrug. And what happens is this group they put on there is sensitive to this liver-specific enzyme, a cytochrome P450, which cleaves that and generates this compound. And even if you're not a chemist, you might see there's remarkable similarity between this, the active drug in the Metabasis series, and Sabet Orem. And the only difference being that this is a phosphonic acid versus a carboxylic acid. And even though that's a very small difference, this compound is about 2 orders of magnitude less potent and less tight binding to the thyroid hormone receptor as Sabet Orem is. So the one thing about the thyroid hormone receptor is you have to get the atoms exactly right to get high affinity interactions and high potency that you'd like to have for your drug. And then theirs also is liver targeted. And then this is a newcomer, which is the only one I've seen so far. It's clinical stage that doesn't have some kind of an acid negative charge at this end. And this is Madrigal, which is the name of the company. And they just recently reported phase 1 data. So let me show you quickly a little bit of the phase 1 data that's been done for this so you can get an idea for how these drugs behave in people and what the issues might be. And I'll start with the study with Sabet Orem that Quatrex ran. They ran a number of phase 1 studies, and this is their first multiple ascending dose study, which was 14 days of treatment followed by a follow-up. And one of the advantages, I guess you could say, of cholesterol-lowering medication drug development is you can get your therapeutic proof of concept from a phase 1 in a 14 or a 28-day study, because all you're doing is measuring the lowering of cholesterol in serum. You're not looking for a clinical endpoint or anything like that. And that's about the only advantage of pursuing cholesterol-lowering medications, I tell you, versus other possible ones. But you do see that. And so that's how they designed this study. And what you can see from this is that what you should get from this is that Sabet Orem and a proterome are very potent drugs. So the doses being studied here are 10 to 100 micrograms per subject per day, one oral administration. So very, very potent. Hormone replacement-like doses, hormone-like potency is what is seen. And you see great cholesterol lowering with Sabet Orem. We actually see it with pretty much all the Thyromimetics and all through the animal studies. That was what we saw also. So dose-dependent lowering of cholesterol. And these are in healthy individuals that don't have elevated or enriched LDL cholesterol. So very, very potent and effective cholesterol lowering, maxing in about 14 days. And then let's look at some of the safety parameters that we might be worried about. Heart rate, you can see over this, that would be the first thing you'd worry about. So these patients are monitored with telemetry monitoring all through the study, giving readouts on heart rate. And there were gratifyingly no effects on heart rate. These two light gray lines are the reference values, and there's no penetration of that. So no effect on heart rate. TSH, the thyroid pituitary axis, we don't want to perturb that. And what you can see is through all of the doses studied, there's no penetration of the lower level of normal of TSH. And remember, you'd think that these patients would be hyperthyroid. You're giving them a thyromimetics. You'd expect TSH to go down because the gland and the axis would interpret that as an excess of thyroid hormone. And even though you see it maybe trending down at this highest dose, you never see it going below the reference range. And that's true, actually, as you'll see for all of the thyromimetics. And the one aspect of the axis or thyroid status that does change with Sabet Orem and changes with all of them, actually, is this T4, circulating T4 measured out of the blood. And I'm showing you free T4, but it actually happens with total T4 as well. The T4 is mostly bound by a carrier protein, so you can either look at free or total T4. And what you see is that at the highest doses with Sabet Orem, it penetrates below that lowest level of normal. Now, at this dose of 30 micrograms, you get about 40% cholesterol lowering. So there are doses where you get the therapeutic effect and no effect on this lowering of T4, but at the higher doses, this is what you see. And let's skip to the Madrigal compound. This is the thyromimetic that's the newest one, published in 2013. In the back, you may not be able to see this, but everything else is the same pretty much as Sabet Orem, but what is different is these doses are substantially higher. So their dose range was 5 milligrams to 200 milligrams versus the 10 to 100 micrograms we use for Sabet Orem. And the reason for that is that they've changed this part of the molecule that is required for high potency. And that part they changed gives them better liver distribution, which is what they wanted for cholesterol lowering, but it alters the potency in an undesirable way. Nevertheless, they see good dose-dependent cholesterol lowering, but then no effect on TSH, no effect on T3, but a lowering at the higher doses of this free T4. And that's a theme that I think is pretty much constant for all of these clinical stage agents, and something that people are still wondering the ramifications of. So the last one I'm going to show you is a proterome. This is the one that made it the furthest, the CaroBio compound, KB2115. And again, its doses are hormone-like. This is actually this is data from a published phase 2. They did 2 phase 2 studies, and this one had 98 patients, and the other one that was reported in the New England Journal of Medicine down there had 300 patients. And they were followed for 90 days or 3 months on dose. And so what you see again is for between the 2 doses, equal lowering of LDL cholesterol for and also apolipoprotein 100, B100, which is simply another way of expressing LDL cholesterol lowering. So you see good lowering that goes back to is dose dependent because it goes back to normal when you remove the dose. And then if you look at the axis, they see the same things that I've showed you, although maybe to a greater extent with their agent. No effect on TSH, no effect on T3, but total and free T4, both of these are depressed below the reference range and then recover to normal. So it's clearly a drug-induced effect. And there's lots of theories about why this happens. It's an unusual perturbation of the axis. The people haven't seen this before. Importantly, there's no clinical evidence of hypothyroidism. There are no hypothyroid symptoms. There are no hyperthyroid symptoms either. So no clinical evidence of that, but you do get this change from ideality. And in spite of this, and for a different reason, the clinical development of this was stopped in 2012. They initiated a phase 3 study that would have hopefully have been their approving study in familial hypercholesterolemic individuals, the heterozygous ones, where there's many more of them. These are patients that have a defective LDL receptor, or in case of the heterozygous, they have one allele that's defective. And they don't typically respond to statins or the other cholesterol-lowering medications as well. And so they started that, and then also at the same time started chronic toxicology, which you need for this indication of cholesterol lowering in dogs. And after 1 year of dosing in dogs, found a cartilage abnormality in the dogs, which is still perplexing to the thyroid community at least. And that actually forced them to stop development, because this was done in Europe, and the EMA asked them to figure out what was going on with this cartilage thing. And this is a small company, and they simply didn't have the resources to do that. And so they couldn't move forward. And so, of course, the first thing you do is ask, is this a thyroid mechanism-based thing? Do hyperthyroid patients have cartilage problems or joint problems? The answer is no, that they don't typically have that. And so it's not clear at this point if that was a prodrome-specific off-target tox or a dog-specific type of thing. It's just not known. But it did lead to that program grinding to a halt. And so just to wrap up, what are the properties that you would want in a thyromimetic for adrenoleukodystrophy? And these are the ones I think, anyway, that are most important. Once daily treatment, just for compliance and convenience, preferably oral. All of these agents are orally active, so that's not an issue. You also want minimal side effects from the hyperthyroid, or manageable, I guess you could say, side effects from the thyroid hormone axis. You know, this T4 thing I showed you, you'd want whatever that might do over a long-term treatment to be minimal, or possibly correctable with another agent if it is a problem. And then the other really important thing, though, I think, is you need adequate CNS distribution, which was discussed in the last lecture. And this, of course, means blood-brain barrier permeability. And this is actually a problem for most of these agents. And the reason is they have this, like I told you, you need this carboxylic acid right here to have good potency. And a carboxylic acid is something you would stick on a drug to make it not get into the CNS, because it completely inhibits passive diffusion across the blood-brain barrier. And so the thing that is required for target engagement, or most potent target engagement, is for the most part a problem. But oddly enough, it is not a problem. We don't understand why this is exactly yet, but it is not a problem for Sabet Orem. Sabet Orem, within this class of clinical stage compounds, enjoys good CNS distribution. And I'll show you the data for this, and others have shown this. And we don't exactly know why this is, but all of these others have been reported to have minimal or no distribution to the central nervous system. And the original report where we showed this was back when we were working on just characterizing it in 2000, and we noticed that there was uptake into the brain, and we reported it, of course, but we kind of were quiet about it because we were trying. You don't want brain uptake or CNS distribution for a cholesterol-lowering agent that works in the liver, because it's not going to do anything beneficial in the brain, and it can only lead to possible problems. And so it's really not something that you'd want, but this drug happens to have it. And we've now, since getting interested in ALD, studied this in more detail. And these are actually data, unpublished data from mice, where we have a LC tandem mass spectrometry assay for Sabet Orem. And we take mice and dose them and take time points, just a standard PK ADME type of progress curve over 8 hours. The half-life in mice, by the way, of Sabet Orem is about an hour and a half. And so at 8 hours here, there's barely any measurable Sabet Orem left. And what you can see, the blue is plasma, and the red is brain, whole brain homogenates. And so you get good brain uptake, or you can actually measure it through a lot of this time course. And the important parameter really for CNS drugs is the exposure, or the area under the curve in the brain divided by the area under the curve in the serum, because this is telling you from the bioavailable dose that gets into the blood how much of the drug is actually making it into the central nervous system. And for what we measure in our assay is anywhere from 0.16 to 0.3 with that ratio. And the way to think about that is about a quarter to maybe even a third of the dose of Sabet Orem given systemically is making it into the brain, which is actually quite a substantial amount. And if you look at all approved CNS drugs, antidepressants, all the things that we know work in the central nervous system, the range of blood to brain to plasma ratios ranges from about 0.1 all the way up to 1. And so this is higher than it should be for sure, and within the range that you see with drugs in the central nervous system used in the clinic. Others have noticed this too, and what they typically do is if they're interested in developing cholesterol-lowering medications like this Japanese group was, they'll use GC1 as an example of a drug that gets into the brain, and then they'll show their data showing that they get no brain uptake. so like I say, this community hasn't really thought about ALD very much, and so where I think you would want CNS uptake. And so you can see this is a study done in rats by this group. They're dosing orally, and you can see good uptake at the 4-hour time point, a ratio of brain to plasma ratio of about 0.23 or so. And then over the long term, a sustained, retention of the drug, which we've also seen in the brain, because for the same reasons, or maybe for different reasons, that drugs have going into the brain, if it's difficult, getting back out seems to be difficult as well. So there seems to be the sustained or longer half-life of it, resident time in the brain, than we would have expected. And so this is really interesting, I think, why this drug gets into the brain, because as I mentioned, this functional group right here pretty much inhibits passive diffusion across the blood-brain barrier, is known to do that in many other agents. And for a long time, it was thought, until the last few years really, that thyroid hormone, which is very lipophilic, got into the brain and did what it does in the brain by passive diffusion. Because of its lipophilic nature, it would cross membranes by passive diffusion. But about 5 years ago, a rare disease, and this gets back to the value of rare diseases and discovering new biology, a rare disease called Allan-Herndon-Dudley syndrome was reported. And what that disease, the patients with that disease is a genetic disease. The patients with that disease have neurological deficits, but in addition, they have thyroid hormone axis abnormalities. But it wasn't clear, but the receptors are okay. It wasn't clear what the problem was. And what they found when they looked for the mutation is it's in this gene right here, at this transporter, MCT8, which is now widely in the community viewed as the active transporter that causes T4 and T3 to cross the blood-brain barrier from plasma and do the job that it does in the brain. And so it's possible, my guess is, Sabet Orem might be co-opting that transporter or using another one. And this is an interesting area of investigation that we're pursuing in my laboratory, trying to figure out what is special about it that allows it to access the central nervous system. So I'm going to conclude now. This is my last slide. These are potent agents, orally active, have all the drug-like properties that you'd want. They're clinical stage also. The thing to worry about, I think, is the axis, or one of the things to worry about besides all the normal things you worry about in development. The special things are these thyroid axis-related side effects, and they would obviously have to be monitored along with everything else, but specifically monitored in any clinical development. Thyromimetics can distribute to the CNS, but we don't really understand how to make them do that yet. We have one example where they do from systemic dosing. And the other thing is thyromimetics, because of their mechanisms, that I think could in theory work for all of the phenotypes of ALD. And so thank you again for the invitation. And if I have time, I'll take questions.
That was great, Tom. So just maybe to tie some of your mechanistic thoughts with the earlier speakers, and forgive me if I've missed this, do we know anything about the effects of these thyromimetics upon microglial function or oxidative stress in general, and has that been measured with this compound?
In the brain, you mean, or in vivo? No, not to my knowledge. I haven't done that. That's on the list of things we want to do, but no, we have not. And actually, even more importantly, or more academically, with thyroid hormone first. But there's literature. Microglia maturation involves thyroid hormone action, and oligodendrocyte maturation does as well. I don't think there's a connection, or I don't know of one with oxidative stress. But anyway.
All right. Okay. I had a very similar question thinking about thyroid hormone and oxidative stress. Since you opened with the slide that thyroid hormone increases your metabolism, and there's this hypothesis in ALD that you have sick mitochondria. So if you treat someone with a drug that accelerates mitochondria and creates more redox, I'd be worried that you could actually make the disease worse because you already have sick mitochondria.
Yeah. So right. So that again gets to. That is like the muscle wasting, weight loss aspect of thyroid hormone treatment. And in the Proterome study, which went 3 months, there was no effect on weight loss or any of those kinds of measures that you'd expect increases in metabolic rate. There was nothing that showed that. And so I think that there will be a window there, a dose window there. But of course, it depends on what dose you need for therapeutic effect with ALD. But that would I would say, fall into a thyroid-specific side effect that would have to be monitored. Yeah.
Thank you very much Well, thank you very much for this excellent historical overview. I have one question. I have not seen the direct link between the treatment with Sabet Orem and an effect of varome chain fatty acids.
I didn't catch that. What?
You didn't show the direct link, connection between treatment of Sabet Orem and a direct effect on varome chain fatty acids.
No.
There's data out there showing an effect on ABCD2.
Right.
And we assume that an introduction of ABCD2 corrects varome chain fatty acids.
Right.
It has been shown for other drugs, but there is no data there showing that this works. And it is technically a very simple experiment. So are you doing that?
We've done it, and it's been done, and I just didn't want to show it because I don't want to make this as I said at the beginning, I wanted to keep, and it's not published, I wanted to make this a academic talk and not a Neurovia talk. But yeah,
but it's a crucial experiment.
No, of course it is, and that's why we did it, and that's why I didn't spend a year establishing a knockout colony in my lab to just show the thyroid hormone effects. So yeah, it does work for that, and
so treatment with Sabet Orem in the ABCD1 knockout mouse?
You get lowering of VLCFAs, yeah.
Okay, that's important.
Some people don't think so, but some people always want what you don't have. So,
have you seen the recent Italian study of the oil of citrus fruits, one I've never heard of, dramatically reducing cholesterol and triglycerides? It was placebo-controlled and blinded, but you'd have to look at thyroid and liver effects.
No, I didn't see that, but there are a lot of things now that lower cholesterol, and in fact, there are lots of good drugs that are approved that are pretty darn safe that do it also, and so. Which is one of the problems that this whole program or effort ran into. Is there are good ways to lower cholesterol, and I wouldn't be surprised if
what's relevant to your work?
Yeah, if there's a thyroid hormone, a citrus. My own guess with what you've just said is that there's no involvement of the thyroid hormone receptor in that because the chemical structure you need to activate thyroid hormone receptor is very restricted, and I don't know of any natural products that do it, and so I would say it's unlikely that there's any involvement. But there are many other signaling pathways that converge on cholesterol homeostasis, so it's almost certainly one of those.
Great. Thanks so much, Tom. That was a great round of applause. Thank you.